Ready?
Let's start. A very good morning to one and all. Now we are going to see about the human chromosomes, and as we all know that human chromosomes plays an important part in our life, being chromosomes and genetics plays a wonderful part in our life, like they carry the genetic information that was passed from our mother and father and to us also to make us totally unique. Here I am Dr. M. Vimaladevi, Assistant Professor, Department of Zoology, Velalar College for Women, Erode, is going to give a brief account on human chromosomes and the massive part it plays in our life. So, what we need, what's the necessary for the need for human chromosomes to study? That is, it carries a major part in our genetics and disease identification for each and every person, and hereditary anomalies which we are being happened in our age old factors. So if we take this genetic studies, all of our cells have the capability to give rise to new cells, and then they are being encoded with informations regarding the living cells which are passed from our generation to generation. That is, their information can be coded in the hereditary material of the cell. So during cell division, it is very essential that DNA remains intact and evenly distributed among the cells. So chromosomes plays a key part in the process that ensures DNA is accurately copied and distributed in the vast majority of cell division. When we come into disease identification, how come the chromosomes plays an important part? See, when a chromosome is abnormal, it can cause health problems in our body. So there are special tests like chromosomal studies to see what the type of problem a person has, and cytogenetics is the study of chromosomes and their role of chromosomes in the hereditary. And for that we are going to see about the karyotyping, which is a test that evaluates the number and structure of a person's chromosomes in order to detect the anomalies. And when we take the hereditary anomalies, chromosomes contain vast majority of genetic information of an organism, as we already see, and it is contained with the genes or DNA, which is contained within the chromosome, is a long series of nucleotides that are organized into genes, and it helps to inherit the characters from parents to offsprings. And what are all the defects which our parents or our grandparents have, it may be heritable, so that we can be able to see with the help of, or we can able to detect with the help of the chromosome studies. And these are all some of the need for chromosomal studies and the need to evaluate or need to find out what chromosomes is responsible for what allosome or autosomal type of diseases each and every persons have. And next we can able to find out the chromosomal types with the help of this slide. That is, in case of the chromosome types, we see that is there are some autosomes and allosomes, two types, and normally the human beings have 46 or 23 pairs of chromosomes which are diploid in nature. And when we take the sex differentiation with the help of chromosomal complement, females have this type of chromosomal thing 46XX and 46XY for males. And if we take the chromosomal complement of female 47XX plus 21, it may be due to chromosomal disorder, and in case of males also 47XY plus 21, it may be due to chromosomal disorder like Down syndrome. And naturally allosomes are meant for sex chromosomal, and autosomes, which is only responsible for growth or physiological functions of the body. And when we take this chromosomes, their structure and nature, German biologist Walter Fleming in the year 1880s revealed that during cell division, the nuclear material organized themselves into visible thread-like structures, which we name as chromosomes, which stains deep with basic dyes. So the term actually chromosome was coined by Walder in 1888, and it's a colored and soma, that is chrome is colored and soma is body, hence they are called colored bodies, and can be defined as higher order organized arrangement of DNA and proteins. and it contains many genes or the hereditary units, regulatory elements and other nucleotide sequences. Also, chromosomes vary both in number and structure among the organisms, and Sutton and Boveri in the year 1902 suggested that chromosomes are the physical structures which act as messengers of heredity, and the length of course from 4 to 6 mu for each and every chromosomes. And with respect to the chemical constituents, that is, it comprises of DNA, RNA, histones which are basic in nature, and non-histone proteins which are acidic in nature. And when we take this DNA, which is the most important chemical component of this chromosome, and it plays a key role in controlling heredity, and also this DNA is usually expressed in C values. And also when come to this histones, we can be able to see that is or basic proteins enriched with basic arginine proteins like arginine and lysine. And there are several 5 types of histones like H1, H2A, H2B, H3 and H4, which are very similar among different species of eukaryotes and have been highly conserved through evolution. And when we take these non-histone proteins, the chromatin comprises of many different types of non-histone proteins which are involved and arranged in several types of activities like DNA replication and gene expression. And also they are being different among different types of tissues of the same organism. So when we come into the next thing, that is a chromosome, see, this is the structure of a chromosome and the various components present in one chromosome, that is 2 identical chromatids, 1 P arm, 1 Q arm, that is P arm is short arm structure, Q arm is long arm structure. Then they are being joined by connected by a bridge called centromere, that is constricted point of the chromosome. And DNA molecule which is of long string, the DNA molecule formed into a compact structure by proteins called histones. And apart from the telomere, secondary constriction and satellite, and chromomeres. So if we take this chromatid, these two are sister chromatids which is present in one chromosome, and during cell division this chromosome is divided into 2 identical half strands joined by the centromere. And a chromatid is each half of the chromosome is joined, contains DNA and separates at anaphase to form a separate chromosome. So both the chromatids are attached to each other by the centromere. And we take this chromatin molecule which is in the chromosome is composed of DNA and proteins. So when we take secondary constriction, generally present for the molecular organization, and telomere, which is present in the terminal region of each side of the chromosome, and chromomeres or thread bead-like structures present on threads or present as threads or chromonema. So they are arranged in a row along the length of chromonema, and the number of chromosomes is constant, and it is responsible for carrying the genes during cell division to the next generation. And matrix, another one, that is a pellicle which is present in the membrane surrounding each of the chromosomes, is a jelly-like substance present inside and formed of non-genetic materials. And when we see these functions of the chromosomes, normally there are some basic functions which like that is genetic information of living organisms, replication, cell division will be carried out by these chromosomes, and which are essential for growth, survival and reproduction of the organisms. And also these histone proteins and other proteins which cover the chromosomes protect it from chemical enzymes and physical forces which is present in the plasma. And thus chromosomes also perform the function of protecting the genetic material, that is DNA, from being damaged during the process of cell division. And during cell division, spindle fibers attached to the centromeres contract and perform an important function. So the contraction of the centromeres of chromosomes ensures precise distribution of DNA, genetic material, to the daughter nuclei. And of course, the activation and deactivation or contraction of chromosomes may be done with the help of these histone proteins, that is DNA coils around this protein. And also spindle fibers attached to the centromere help in the movement of the chromosome during the cell division, and chromosomes thus have thousands of genes precisely code for multiple proteins present inside the body. And this is a type of thing which is we segregate the chromosomes through karyotyping, like according to the centromeric position group from group A to group G, that is chromosome number 1 to 3 is of largest and metacentric, and group B has chromosomes 4, 5, that is medium sized and large and submetacentric. And if we take this group chromosomes under group C, it is often 6, 7, 8, 9, 10, 11, 12, that is of medium and submetacentric. And group D has chromosomes 13, 14, 15 has medium, acrocentric and satellite chromosome. And group E has 16, 17, 18 medium, short, metacentric or submetacentric chromosomes. And F has 19, 20 short, metacentric, and G has 21, 22 smallest metacentric Y and X chromosome. And if you see a small thing like syndromes which is being due to the autosomal anomalies, a Down syndrome also termed as trisomy 21, and this is when a baby is born with an extra copy of their 21st chromosome in some of their cells. They have an extra chromosome in the 21st chromosome, that is addition in the 21st chromosome. And why it is an important or a thing to understand this means chromosome missegregation can lead to a jumbling set of chromosomes where the wrong pieces of the genome are stuck to the wrong place. So instead of 46, they may have 46 plus 21. So these Down syndrome patients are being short height, severe mental deficiency with decline in IQ, with age, brachycephaly with flat face and occiput flat and low nasal bridge, and also simian crease may be present. And there's clinodactyly with fifth digit in the fingers and malformed large ears as you see in the picture, and epicanthal folds of the eyes you see, and then brush field spots in the iris, and renal anomalies, several renal anomalies like kidney diseases, and prominent and protruding tongue will be there. And this is one of the autosomal syndrome which the child faced due to this trisomy 21. And these are some of the pictorial representation of the Down syndrome. There you see the triplication, that is trisomy in the 21st chromosome, and the protruding tongue, and simian crease, iris defended, and then normal palm when compared to simian crease palm has been shown in the chromosome. And of course thus chromosomes plays an important role in the heredity of an family of an or an individual, and thus it is necessary to find out our hereditary patterns or the pedigree patterns which describes whether any anomalies or syndromes or chromosomal defects which are being inherited from our forefathers or parents, and this we can able to somehow manage through genetic counseling. And nowadays lots of novel treatments are available like gene therapy. Through that we can able to manage those inherited disorders, and thus it is quite important to understand what type of inherited disorders if at all a person is having through pedigree analysis. And for that we have been having specialists like genetic counselors, genetic doctors, and then so likewise there are several therapies available now in the current scenario. So with this, I wind up this short account on chromosomes and types of chromosomes, and with a brief introduction about one of the disorder. And let's see the detailed syndromes which may be possible due to the chromosomal anomalies in the next session. Thank you one and all, once for all. Thank you.